Good evening, ma'am.
Good evening, sir.
Okay, thank you. Shall I start, ma'am?
Yes, sir. You can start now, sir.
Yes, ma'am. Thank you, ma'am. Good evening, my dear students. Once again, you are all welcome to the biological session. Last class, what we have discussed, already you know about. So, what is meant by polyploidy? Okay, and what is meant by plant introductions? And what are the two types of introduction is there? One is called as indigenous collection and exotic collections. Already we discussed. Indigenous means within the country or same country. Okay. And the exotic means it's outside the country. That things also we discuss, and also what are the examples they have given. Okay. That things also very, very important, my dear students. So today we are going to discuss in these sessions. So what is by polyploidy? Last class we saw it. What is by autopolyploidy? Ploidy is nothing but number of chromosome increases, isn't it? Polyploidy means more than one. Autopolyploidy means what is that? Within the same species. Okay, my dear. Autopolyploidy. See, the autopolyploidy is produced spontaneously in nature, naturally occurring. Ploidy means what is that? You know, already we discussed. Take an examples. Addition of chromosome, isn't it? Addition of chromosomes. Already we know about. For example, so N is denoted as haploid, 2N denoted as diploid. Suppose you are adding 2N plus n, that is equal to triploid. Suppose 2n plus 2n, what is that? Tetraploid, isn't it? So these two, so more than 2, it is considered as a polyploidy. More than 2n, it's considered as a polyploidy. Already we know about. Suppose if it is occur in naturally, okay, so that is called as autopolyploidy. Okay, it can be induced in the relatively high frequencies by colchicine. What is the meaning of frequencies? Frequency is nothing but the nitrogen bases. Okay, nitrogen bases present in the gene. Okay, always called as genetic material. Which one is the material? Already in our world. It's a chromosome, particularly in our world, gene or chromosome, isn't it? So adenine, guanine, cytosine, and thymine. These are the component. Okay. The colchicine, what they will do, it is very, very important. Alkaloids. Yesterday, I told last class we discussed this part. Alkaloid substances to prevent the formation of the spindle apparatus during mitosis. What is the main uses of this one? You know, mostly they use the colchicine. Okay, it is one of the alkaloid substances or alkaloid chemical substances that prevent the formation of spindle formations. What is the use of spindle formation? Already you know about mitosis. What is mitosis? Already you discussed. Okay, what is the meaning of mitosis? Mitosis is nothing but the cell division which occurs in the somatic cell, isn't it? So as a result, what happen? You know, you will get 2. So for example, so this is considered a single cell, isn't it? Single cell. So when the cell is undergoes mitotic cell division, what happen? How it will be? So single cell divided to give 2 cells, isn't it, my dear? How it will be possible? Already in our world, there is a chromosome is there. Okay. The chromosome also multiplication takes place. What are the phases? The mitotic already in our base. Prophase, anaphase, metaphase, telophase, diakinesis, isn't it? So these are the things. But here, so the colchicine is a chemical substance. Colchicine. Okay, colchicine. See here. Okay, colchicine. So this is a chemical substance, otherwise called as alkaloids. Okay, the alkaloid, what they will do, you know, it prevent the, just imagine, prevent the spindle formation, spindle formation. You know about that. Suppose the single cell is converted into 2 cell by the process is called as mitotic division, isn't it? During this division, the chromosome is also divided. Cytoplasm also division takes place. Cytokinesis and karyokinesis takes place, isn't it, my dear? How it will be divided now? With the help of the spindle fiber. Already you know about. Take an example like this, isn't it? Take an example like this. See, consider this one is considered as a chromosome. Okay, just imagine this is also one of the chromosomes, and another one is also a chromosomes. During the cell divisions, multiplication takes place. Here you know very about the, what is that? Centriome. So what happened? Automatically the cell division. This is considered a, okay, spindle formation. This is spindle fiber. This is called a spindle fiber. Okay. So when it undergo the division process, what happened? Oh, see, automatically they can migrate towards the poles. Okay. So as a result, what happened? The result will be like this. Okay, they can migrate towards the pole, isn't it? So they can migrate towards the opposite pole. They can migrate like this. Center, this is a center metaphase to here. So during this, the interconnected with the spindle fiber, it can be prevented. It can be prevented by the colchicine alkaloids. So as a result, what happens? No, this the cell division, it will not occur. That is the reasons. So this is mostly used in the cancer cells. What is by cancer? Cancer is nothing. It is also one of the mitotic cell division. Mitotic cell division. Okay. And rapid cell division, isn't it, my dear? Cell division. That means uncontrolled growth of cell division. It is also occurred due to the presence of which one, the spindle fibers. But when you are applying this one, what happens? Automatically the spindle fiber formation, it not takes place. So as a result, the single cell converted into double cell, it is not possible. So likewise, the cancer cell, the multiplication is also stunted due to the application of colchicine. What happened? The single cell, that spindle fibers formation completely stopped. As a result, the mitotic cell division, it will not happen. So the cell converted into 2 into 4, 4 into 8, it multiplication rapidly, formation is also stunted. Isn't it, my dear? So that is the process also given here. That is the process. Okay, my dear. But the next one. See this one. The colchicine spindle apparatus of matters. Sometimes an autopolyploidy has a succeed as a variety of example on autotrophic variety of tea in India. So this is also very, very important. Let us discuss. Most of the present day crops such as tomato, grapes, watermelon, banana, pineapple are autopolyploidy. They ask the questions. Okay, they ask the question, this part. So given examples of which one of the following is an examples of autopolyploidy. Sometimes they may ask the questions, which one of the following is not related to the autopolyploidy. Like that, they may ask the question, whether it may be a positive aspect, whether it may be a negative aspect, they may ask the question. But the next one, allopolyploidy. It is also another category. It is also another type of ploidy. Allopolyploidy arises as a result of interspecific hybridization. Interspecific. What is the meaning of interspecific? Means different. Different. Okay. Different. Okay, just don't forget. See the first. 2 different species. Take an example. 2 different species are hybridized. To place the F1, the F1 is usually highly sterile in nature. This is also an important question in NEET point of view. Due to the allopolyploidy, the F1 generation, it will be look like a fertile, sterile. Okay, like that they may ask the questions. Match the following assessment reasoning. So also they may ask the question, which part? F1 generation, phylyl 1. Phylyl 1 is nothing but the first generation. Okay, therefore, in the second step of the chromosome, number of the F1 is doubled. This is also an important one. Okay, this is also an important NEET questions. Allopolyploidy, the F1 is sterile in nature. After the second step, what happened? It will be double in nature. For example, 2 means 4. That's why it is considered, it is known as ploidy. Okay, number of chromosomes, it will be increases. So ploidy. The resulting allopolyploidy is usually at least partially fertile and forms a new species. So this is also called as partially fertile. So one more name is called as allopolyploidy is also called as, what is that? Partially fertile. Okay, my dear. So this is very, very important. How it will be already told. So what about the F1 generation? F1 generation is completely sterile. So these are the 2 important questions arises. They may ask the question NEET point of view, my dear. Okay, this is also a NEET question. Okay, many allopolyploidy have been produced in nature. Okay, it is also naturally occurring. Some of which have succeeded as a crop such as wheat, oats, tobacco, cotton, and pomoto. What is the meaning of pomoto? Pomoto means potato as well as the tomato, the combination of two plants, same species, Solanaceae family, potato and tomato. I have to take which one is the first part of the first species and the last part of the second species. Tomato. Okay, my dear. So this is very, very important, my dear student. This is also a Allopolyploidy. And humans have produced a new Allopolyploidy crop called triticale. This is very, very important in the year 2017, I think. Okay, the question asked. The triticale, okay, triticale is the following manner. Allotetraploidy, wheat, triticum, what is that? Durgidum. So triticum durgidum was hybridized with the Secale Cyril ray plant. See here, these are the two combinations here. The wheat is also combined with the ray. See, so wheat combined with the ray plant, white with ray. Okay, so called Secale Cyril. It is a diploid. This one is called as what is it? Allotetraploid. Tetraploid, what does it mean? 4 in nature here. This one is called as what is the diploid nature, 2 in nature. Okay, triticale is cultivated in some area of Punjab and the hilly region of the country. Okay, so finally we'll get the which one? Triticale is the part. Triticale means the first one, part 1 is the, and the last part of the second species. The first name of the name is here. So 2 name is the genus name as well as the species name, isn't it? But triticale they have given is a common name, isn't it? This is the variety. This is the variety of 2 species, but same family. But the name is called as triticale. Triticale means triticum, the first 3 letters of the genus name. And Secale and Cyricale. So the last name of the genus name or species, whatever we can take these 2 or 3. Particularly, you have to take genus name as well as a species name. Here we have to take genus, isn't it? Genus name. Here you have to take the species name. Okay. So combined together, you will get triticale. Okay. Mostly they cultivated in the which area? The Punjab wheat. Okay. Wheat mostly. Which is the wheat bowls of India, Punjab only, isn't it, my dear? So don't forget, my dear, this is also a very, very important question. And haploid nature. What is my haploid? Is nothing but the single N nature. Okay. What is by haploid? See that? Haploid. Haploid, nothing but the single. A haploid has only 1 copy of each chromosome. See this one? 1 copy of each chromosome. And is highly sterile in nature. Highly sterile in nature. Haploid plants are produced in nature at a low frequency. But in many species, they can produce in high frequency by culturing their anthers and ovaries. Normally, haploid plants are sterile. Okay, good. And haploid plant produce in nature at a low frequency only. Very less nature, less only. But many species, they can produce in high frequency by culturing their anther and ovaries. Haploids are used for producing homozygous lines in about 2 years. Homozygous. What is the meaning of homozygous? Both the chromosomes are same. See the capital T, small t, small t. Isn't it, my dear? About it will take 2 years. So these are the important. Okay, my dear. Go to the next one. Mutation breeding. What is my mutation? What is my breeding? Mutation is the process by which genetic variations are created. Okay, it may be artificial or manual. This one is called as mutation, is nothing but the factor is that there are so many factors, particularly physical factor and chemical factor and also biological factor. Isn't it? There are so many factors there. Mutation is the process which genetically variation created mutation. Okay, otherwise called as mutation is nothing but the sudden changes. Sudden changes occurs in where? Chromosomal level. Where when there is a change occurs in the chromosomal level, automatically it may be transferred from the one generation to another generation, isn't it, my dear? So that is very, very important. See here, it is possible to induce a mutation artificially, that is man-made, through use of chemical radiations. How it will be? Due to the UV rays. Okay, okay, and also UV rays and also X-rays. Okay, X-rays, just imagine. And also gamma rays. Okay, my dear. So these are the very, very important things. How it will be takes place? By the action of which one, you know? See that one? Mutations artificially through use of chemical radiation such as the gamma radiations. That's why I told you. Gamma radiations, this one. And selecting and using the plant that have a desirable changes as a source of breeding. This process is called as mutation breeding. Okay, so this is very, very important. After the mutation, the breeding is also done with the help of the gamma radiation. Such type of process is called as mutation breeding. You can imagine like that. Okay, go to the next one. In India, more than 200 variety. Note it down, my dear students. This is very, very important, my dear. This is very, very. The 200 varieties have been developed through the mutation breeding. 200 variety. How many varieties they have been introduced in India? Like that, they may ask the questions. Example they have again, monk beans, resistant to yellow mosaic virus, YMV. Already you know about TMV, isn't it? Tobacco mosaic virus. And cauliflower mosaic virus. These are the two things already you know about. But here they have given us a yellow mosaic virus mostly present in the monk beans. Tobacco means tobacco plant. Cauliflower means cauliflower plant. But yellow mosaic virus and powdery mildew was introduced by mutation. What is mildew? Powdery. There is a leaf is there. Just imagine which leaf they are told. The yellow mosaic virus. Monk beans. Okay, that leaves, it contain, what is that, you know, the water, that is white color powder nature. Okay, in the upper part. Okay, there is a white color patches. Okay, made us. So mosaic structures. Mosaic means what is that? More than one colors within the same places look like a mosaic. Okay, made it. It's like this. This is called as what is that? Powdery mildew. Was introduced by mutation. Next, we will pass on to the next one, my dear students. Mutant alleles are generally receive and produce harmful effects. When mutation always alleles, what is that? Genetically, what happened? Changed that, isn't it? So they will produce what is that? Recessive, general recessive and produce harmful effect. Don't forget, my dear students. Recessive effect. Only about 0.1% of the mutant alleles are beneficial only. How many percentage the beneficials due to the mutant breeding? Like that, they may ask the question. This is also sometimes they may ask the question, NEET point of view. Okay, media. So the beneficial one. In view of the extremely low frequency of desirable mutations, spontaneous mutation cannot form the basis of the breeding program. But spontaneous mutations are the source of all the genetic variations we see in the biological world today. Okay, this is naturally occurring. What is that? Naturally occurring. What is that? Mutation. That is called as spontaneous mutation. Now we are seeing so many mutation changes within the organism, whether plants or animal, whatever it may be. How it will happen, you know, due to the spontaneously occur mutation. We are seeing today world, biological today world. That is very, very important. Okay, media. So go to the next one. Some examples of mutation breedings. So already we discussed this point. Anyhow, just to note it down, this is also very, very important. Take an examples of rice, Jagannath. Okay, see that automata. So 2. And wheat, Sarbadi sonara. Already I told you, M.S. Swaminathan. Pusa lerma, where wheat. These are the wheat. Okay, don't forget. They asked the question, Sarbadi sonara is a wheat. This is also belong to the family. Poaceae. Poaceae family. Variety developed from the red grains variety. Okay, 30 and Nora 64 and Lemra Roja 64A respectively. Through the, what is the gamma radiation. So this is the meaning. How it will be, you know? So these are the things. Nora 64 and Lemra Roja 64A. Both are united together, you will get Sarpadi Sonara. Okay. How it will happen? Gamma radiations. So you have to mention Nora 64 plus Lerma Roja 64. Okay, both are what is that? Wheat. Okay, wheat. Wheat, both are wheat family, same family. Okay, here what they are doing now? Gamma rays. Gamma means this rays. Okay, when you are introducing, you will get the new variety. Okay, which one you know? That is called as Sarpadi Sonara and Pusa Lerma. So another one is castor. Already you know it is also oil seed. Okay, oil seed. Fabaceae family. Already you know Aruna. It's a new name. Aruna is a name. Sarbadi Sonara is also name. So Jagannath, Atomita is also 2. 2 is means in number of discovery, like the number of the time, it may be different. Okay, my dear. So these are the importance. Monkombu Sambasiva Swaminathan. What is the meaning of M.S. Swaminathan? So they have given us a Monkombu. See this one. What is that? Monkombu. Okay. Monkombu Sambasivan Swaminathan. Simply they called us a Sambasivam Swaminathan. Simply they called us M.S. Swaminathan. Okay. What is it? M.S. Swaminathan. Okay, my ideas. So this is M and S Swaminathan. Otherwise simply called as MSS. This is also an examination point on NEET. Where it located in Goa or Mumbai. Okay, Mumbai. Where is M.S. Swaminathan laboratory located? Sometimes they may ask the question Mumbai. See here, he was born in Tamil Nadu. He developed short duration, high yielding varieties of rice, including scented basmati. Imagine basmati rice is also. He is also known as known for the development of the concept of the crab. Okay, cafeteria, crabs scheduling, cafeteria and crab scheduling. He initiated collaboration with Dr. Porlock. Is the world green, father of world green revolution, which culminated into the green revolution through introduction of the Mexico variety. Already we discussed this part in the intro part. Of Wheat India, he's also initiated the lab to land. This is the important part. So lab to, what is that? Lab to land cultivations. Land cultivation was done by, who is that? Two scientists is very, very important. One is called as M.S. Swaminathan, another one is called as Porlock. Okay. They did, which one, you know, so many concept development, the development of the concept of the crab development, like that it may be. Cafeteria development and crab scheduling. Okay. So that is also very, very important. And also cultivation from the lab to land, that is called as hardening. That process is called as, what is it, you know, hardening. Just when you are going to study in tissue culture, you can see this point. In the last one, what happened? No, test tube is there. So after the test tube cultivation to transfer to the land cultivation. Okay, that process is called as hardening. Okay, media, don't forget, this is also very, very important. When you are going to study the tissue culture, you can study these points. And many crab plants are now, okay, unable to grow in the wild. They have become completely dependent on human beings for their survival. For example, maize. Because of this, maize is often called, what is that, pembered corn. In many cases, even wild ancestor has become obscure. For example, cabbage, maize, etc. Okay, media. So these are the extra points only. And selection of the self-pollinated crops. How will you select the selection of the self-pollinated crops? It is one of the breeding technology. Excuse me. Self-pollination causes rapid increase of homozygosity. Already I told you. Homozygosity. What is the meaning of homozygosity? For example, capital T, capital T. You will get the capital T and capital T. So again, you are crossing self-pollination. You will get the capital T, capital T. The homozygosity it will be get. So like this. Again and again. How many times? You can put 100 times, you will get capital T only. Okay, that is called as homozygosity. The zygote, it will be only one type only. Homozygous, both are. Okay, only one type. As a result, population of self-pollinated crops are a mix of different homozygous genotype. Okay, so it can be crossed with another homozygous. Take an example, small t, small t. Here you can calculate it. How many times? 100 times. What happened? Only small t only, you will get it. So this homozygosity as well as this homozygous. This is a doll. This is called as dwarf. You can cross it, you will get the heterozygosity. See this one? Selection of such crop, therefore, ultimately isolated the best homozygous genotype present in the population. This genotype may be used as a new variety. Wheat variety PV18 is also example. Kalyan Sona is also one of them. And also HUW4868 is also one variety for wheat. Another one is maize variety, Vijaya and Ganga. Don't forget, my dear student. These 2 points mostly asked in the NEET point of view. Match the following. Ganga is the one of the following. Which one of the following variety of cereals or pulses like that? They may ask the question. This one is called as what is that? Cereals. Maize varieties. Vijaya and Ganga. Okay, media. Don't forget. These are all comes from where, self-pollinated. Homozygosity. Self-pollinated crops are mostly found in which type of zygosity? Homozygosity. Okay. So these are the very, very important question, my dear. And also next one is a selection of cross-pollinated crops. How it will be collected? How will it be collected? Cross-pollinated crops. The population of cross-pollinated crops are heterozygous. Already we know about. For most of their genes. Selection of cross-pollination. See here, take an example. This one is capital T, small t, small t. Okay, this is a homozygosity, isn't it? When you are crossing these two, capital T, small t is you will get. This is called as heterozygous. Heterozygous. They give 2 types of gametes, isn't it? Capital T, small t. Again, cross with the capital T, small t, you will get 4. 1:2:1 ratio. Already you know all about the monohybrid cross. Here, this is also homozygosity. This is also homozygous. This one is called as heterozygous. That things as given here. Most of they are genes. Any decreases in heterozygosity reduce their performance. Selection in such crops aims at increasing the frequency of superior genotype without reducing heterozygosity. Therefore, selection can be continued in the successive generation of cross-pollinated crops. For example, already we studied in outer breeding inseminations. Outer breeding, what is that? What is meaning outer breeding? Sometimes it may be the gametes are isolated from the different plant or different country. Okay, otherwise called as invade from the one country to another country. Like that, imagine here. Capital A is that. This is a homozygous, isn't it? The other country, they may come with capital B, or small b, like this. This is the, what is that? Cross-pollination. So this time what happened, the cross-pollinated crops, the F1 generation, it will be very high. Sometimes it will be high in nature. It's a good. Suppose if it is less, what happened? Suppose you are doing the hybridization up to 10 times. Okay. So the 10 times what happened? The cross-pollinated depression also takes place. Cross-pollinated depressions. That is also very, very important. So the when you are going to select cross-pollinated crops, very careful, my dear. Then only you will get the success of generation. Otherwise, what happens? The inbreeding. What is that? Cross-pollinated breeding depression takes place. It will not get the good result. That is very, very important. Okay, next, clonal selection. It is the process of selecting plant with useful variation for vegetative multiplication. Clonal selection is also very, very important. Okay, selection of plant without disease resistance, tough, that means the climatic resistance and also antibiotic resistance, pest resistance, so many things is there. Okay, so they are all used for the which one, the vegetative multiplication process. Vegetative multiplication means you know what about the propagation is there. And tissue culture is the most important one. Okay, vegetative propagation. For example, some of the plants are caused by the disease, affected by the disease. What happened? They will not produce a good yield. After 10 years, 100 years, the plants also destroyed completely. That time, what they will do? They will regenerate the plantlets in the form of, what is that? Tissue culture, by the process of tissue culture. Okay, media. What is it? Evaluation and release the variety. See, this is one of the other important points only, because in India, the Indian Council of Agriculture Research, where it located, this is also very, very important question. Sometimes they may ask for ICAR, Indian Council of Agriculture Research, located in New Delhi. Don't forget my PUSA. Okay, research is where it is. So like that, they may ask the question. These two questions very, very important. So many times, match the following. Okay, match the following. Sometimes direct question also. Carried out the evaluation. Suppose we are introducing new variety. Who evaluate the plant or that varieties? Means this Indian Council of Agriculture Research Fund is responsible for that one. The materials developed by many breeders are evaluated together at several locations in different agriculture zones. Agri-climatic zones. Take an example, agro-climatic. Don't forget, climatic means different region is there. Take an example. In India, how many climate zone is there? Take an example, north, south, east, west, whatever it may be, different zone is there. The whole country is divided into a agro-climatic zone on the basis of soil and climate conditions. This is also very important, my dear students. How the whole country of India is divided according to their agro-climatic zone, according to their agro-climate. Like that, they may ask the question. Two things is agro means what is that? Soil, nature is depends upon their based of soil and climate conditions. The number of these zones depends on the crop in questions. Each material is evaluated in the single agro-climatic zone for a last 3 years. Each one, it is considered a cultivation up to 3 years. In every evaluation, performance of the new material is compared with that of the existing variety. New material is also compared with the already present varieties. So we then only they come to the conclusion which one is a very good breed, which one is very beneficial to the public, like that they may imagine. Okay, that one approach to the farmer. The breeder of such a materials make a proposal for its release. Release means what is a farmer. Okay, media. Okay, so this is very, very important. This proposal then considered by the variety releases committee, and also the committee also is there. If the proposal is accepted, the new material is given name, and this given name is also the next one. Is released that a new variety that is called as IR. 8. Already you know all about. Now IR 24. So what is the meaning of this one? This one is also both are rice only, isn't it? How it will be introduced? 8 or 24. It's a new variety. These two are new variety. The government notifies each releases. That is very, very important. Evaluation and research of the variety was done by Indian Council of Agriculture Research only. The Indian Council of Agriculture Research located in the New Delhi. Okay, so that is very, very important. And also the how they are, the whole India, how they are classified. That means according to basis of soil and climate conditions only. So how many years they will grow means up to 3 years. What is that? Evaluation is also done. After that, these are the processes. The committee is also accept. Name is also given. Then only they have given a new variety to the public. So multiplication is improved seed. Multiplication of improved seeds. What are the multiplications? Already we did autoploid, polyploid, allopolyploid, that things, isn't it? How it will be improved. The quality of seed is often certified by the agency. When it is done, the seed is called certified seeds. There are so many seeds in the beans. Okay. So many seeds have been given. Beans. Okay. Soya beans, mungu. Okay. And also Bengal gram, black gram, like that. So many things. They are all considered known as Solanaceae family. So how they will be assessed? National Seed Corporation. That is simply called as NSC, National Seed Corporation. This is also NEET question, my dear. Okay, NEET question. Short form. They have given in the short form. You have to expand it. Was established to the streamlined seed production in India. It is continues to play a central role in the seed multiplication. Okay, just to take an example of Switzerland, there is one seed production area is there under the snow. Okay, the world seeds, what are the seeds present in the world? Those seeds are stored in where minus 196 degrees Celsius under which one liquid nitrogen. Okay, liquid nitrogen. Don't forget, my dear student. This is very, very important. Where Switzerland. It's nothing but the playground of Europe. Okay, my dears. Don't forget, these are the very, very important things. See here, inbreeding depressions. This is a very large process, my dear students. When two individuals of the species that are released by the descent and they mated together, it is known as a inbreeding. Okay, inbreeding. Inbreeding is what is that? Two related species also. What is that? Inbreeding. Example. Two individuals of a species only that are related by the descent or mated together, it is known as the inbreeding. Outbreeding means different species in various. The most extreme form of the inbreeding is self-pollination. Only already discussed this part also. Selfing. Inbreeding depressions may be defined as the loss of vigor associated with the inbreeding. This is also very, very important. Why? We are not marriage. Do not marriage our uncle, daughter, or like that, because there are some reason is there. What is that reason? Due to this one. Loss in vigors associated with inbreeding. What is that? Inbreeding depressions. Already told you. Okay. The F1 generation, it will not good. Because suppose the family, it has any diseases, it's surrounding itself, their family only, they will not go out. Okay. So the majority of the cross-pollination crop and also all animal species, so the inbreeding depressions. See, this is a very, very important inbreeding depression. On the other hand, none of the self-pollinated crops show the ill effect in breeding. So this is also another one. The phenotype superiority of the hybrid F1 plant over either of its parents in one or more triads is called as hybrid vigorous or heterosis. The term was coined by Skull. So this is very, very important. Heterosis name was given by the name of the scientist there. What is that? Shull. That is very, very important. S-H-U-L-L. Okay. The most likely explanation for the inbreeding depression is that the dominant alleles of the all genes have beneficial effects. However, the recessive alleles of the most, if not all, genes are deleterious in vigorous degrees. What is the meaning of this one? Just imagine, take an example, some monohybrid. Okay, capital T is there, small t is there. This is the dominant, this is the recessive, isn't it? This one is called as tall, isn't it? Dwarf. Okay. Suppose if it is comes from the same species, means what is you will get capital T, small t, isn't it? This is heterozygous. Again, they will split capital T, small t. So what happened at the self-pollination once you are doing after 100 times? What happened? What is that? The inbreeding depression happened. Crossbreeding means what is just imagine. Just imagine the recessive gene also comes from the other foreign particle, just as a capital G. This is a what is a different breeding is there, but it will not come. It will not happen in the inbreeding depression because it is outbreeding, isn't it? The depression, it will not happen like that. They told you. So during the self-pollination, what happened? The F1 generation, it will be not good. That is the important. However, the recessive alleles of the most, if not all, the genes are deleterious of vigorous degrees. Plants of cross-pollinated species are highly heterozygous. Therefore, they contain recessive allele also most of the genes in heterozygous state. The harmful effect of the recessive alleles as a result will not be expressed. That is very, very important. During the what is that? Inbreeding pollination or self-pollination, what happened? The F1 generation, the recessive genes, they produce harmful effect, isn't it? But at the case of what is that? Outbreeding effect. Outbreeding, otherwise called as what is that? Cross-pollination, what happened? Interbreeding depression, it will not happen because the recessive gene, it will not express harmful effect. When such population is subjected to the inbreeding, there will be an increasing homozygosity. Okay. As a result, many recessive alleles would become homozygous. These recessive alleles will express their harmful effect, causing inbreeding depression. Okay, my dear. So this is very, very important. See that a heterosis. Heterosis is the increasing in growth, size, function. Okay. Yield or other characters in hybrids over those of the parents. Heterosis is essentially the reverse of the inbreeding depressions. Okay. So what is the meaning of, this one? So this is called as parent A. This one is called as parent B. Okay. This is F1 generation. What is the meaning of F1 generation? Just imagine. So this is considered capital A, capital A. This one is considered capital B, capital B. Parent average, this one is F1, isn't it? F1 means what is it? Capital A and capital B is there. Here, heterozygous nature. Heterozygous, isn't it, my dear? Heterozygous nature. Okay. So when what happened, heterozygous condition, it comes, so sorry, here I have to see this one. Okay, just imagine. Sorry, here I have to put a small letter because this is a small b, small b. So here also I have to change it, capital A, small b only. Sorry. So this is the heterozygous, isn't it? Heterozygous. This is called as a recessive gene, recessive. Isn't it recessive? SSIV. Okay, recessive gene. The recessive gene also expressed here, but it will not produce any harmful effect because it is heterozygous nature. Okay, so what happened? The effect of heterosis is what is that? Reversible manner. Reversible. What is that? The effect it will be reduced. That is the important point. Suppose if it is a homozygous, that means self-pollinated. Means what happened? The heterosis, it is not like this. This portion is called as what is heterosis. They still told. Okay, so this is the parent A, parent B. Here the dominant. This one is the recessive. When you are getting the capital A, capital B, this is the recessive conditions, heterosis condition. Okay, my dears, that is all. So in this code, what happened? It will not produce any. Heterosis is essentially the reverse of inbreeding difference. The reverse means what is that? Positive result it will give. That is the reasons. So reverse of inbreeding depression means what is that? Reverse. That means positive. It will not produce negative effect. Negative will harmful effect. Okay. So positive result only they will give during the heterozygous condition in. So when the two unrelated individual, take an example, when two unrelated individual or line are crossed, the performance of F1 hybrid is often superior both to the both the parents. So this is the things. Capital A, small a, small a, capital B, small b, capital C, small c. Here they have given different manner, isn't it? See that? Here small c, here capital C. Small d, capital D. So what about the F1 generation? You take an example here. You have to take only one gametes only, capital A. Here only capital B, small c and small d. Isn't it, my dear? Here, small a, small b, capital C, capital D. You cross with this one. So here too. Yeah. So you will get this one. Capital B, small b. Capital C, small c. Capital D, small d. Again, you are self-pollinating the same process. What happened? The depression happen. That is the reason. See here. For example, F1 hybrid producer crossing the line A and B will superior of the both the parent, isn't it? Both the parent it will be superior. We both are superior nature. This F1 will be superior, isn't it? F1 will be superior because the harmful effect of the A, B, and C and D alleles will be masked by the dominant allele. See, these are the here the recessive allele is the A, B, C, D. Here there is a dominant allele is also present in it, isn't it? The dominant allele express itself, but it masks the recessive alleles. It will not express. Okay, see this. This phenomenon is known as heterosis. Both are present in it, but the expression will be only which one, dominant character only. This is also indicate which one, law of dominant. Okay, media, law of dominant. See that almost all species show heterosis. See here, all the species, AA, see all this. Okay, capital A, small a, capital B, small b. See, all are having the heterosis nature. What is it? Heterosis nature, all are. Okay. So it is profitable in vegetatively propagated plant such as mango, apple, potato, sugarcane. Don't forget, my dear student, this is NEET question. What is my heterosis? What is that? Both alleles, heterozygous condition means capital A, small a. These are the two alleles present in the same chromosome. The dominant alleles only express itself. This small one, recessive allele, it not express themselves. This condition is called as heterosis. Okay. This is very important for the most beneficial or profitable in the vegetatively propagated plants such as mango, apple, potato, and sugarcane, etc. That is very, very important. Okay. See that one. Go to the next one, my dear. Now, application of heterosis, where it will be used. Okay, where it would be used. Increasing the yielding and nutrient value of crab plant. First point is, first of all, commercial purpose only, increasing the yield. For example, 10 mangoes per pouch, per bunch, per bunch. Like that, imagine. The same things also, 25 mangoes per bunch of banana. Okay, likewise. And also increasing the growth rate. That is also another point. Enhancing the meat productions, enhancing the milk yield, egg production. These are all only for the animals. Okay, take an example. Meat production means the weight, increasing their weight. Enhancing the milk, what is the meant? Milk and yield and egg production. This is also a commercial purpose. This is also a commercial oriented. Okay. And then fourth point is improving the performance of various animals. Performance. What is the performance? These are the performance. What is that? Enhance the milk yield and egg production is also performance. Such as a race, and one more is the races of horses and domestic pigeons. Races of horses means what happened in it will be used for the, what is the race? Okay, financially, commercial purposes also. Domestic pigeons, number of pigeon, it will be increases. And production of new combination of colors and scents in the garden flowers. Sometimes what happen, production of new combination means it's a new variety. Okay, colors and scents. Take an example, red and white. What you will get? Pink. This is a new variety. Okay, green and blue. You will get brown. Okay, so these are the new variety. Likewise, you can get it. Okay, my dear. So these are the important points. So these are the application of the heterosis, my dear. And the next one is breeding for nutritional quality also. This is also very, very important. Already we discussed it. Okay. Vitamin A containing rice, carotene rice, vitamin A. Okay. Carotenoid rice. See that one has given here. Nutritional quality of crops refer to the suitability of its produced in human animal nutrients. Similar this one. Okay. A crop produced should provide the optimum nutrition and must not contain an anti-nutritional factor. Okay. Sometimes they may ask the question, breeding for nutritional quality. Okay. Such type of crop, they will not contain anti-nutritional factor. That is very important. Which factor it will not present in it like that. Sometimes the anti-nutritional factor present in the nutritional breeding for nutritional quality. It is also one of the quality or basics like that they may ask the questions. Okay, medias. So go to the next one. Anti-nutritional factor. What is meant by anti-nutritional factors? So we will finish it up to this level, my dear students. See that anti-nutritional factors are those compounds that have adverse effect of animal or human growth and development. They are toxin containing in plant as a defense against excessive grazing of grazing. Okay. Some of the plant, they are having toxin substance. So what is the use of this one? These substances, what happened, used for the prevention of the animal defense against the excessive grazing. See that these are the very, very important mediums. Don't forget this one. Glucosinate present in the Brassica oleracea. Sinalbin in a various member of Brassica. Cyanogenic glycosides in almonds. Neurotoxin cyanoalanine is Lathrace sativa, is one of the beans. Cassery, that is name called cassery beans. Regular use of seeds, Lathrace sativa, cassery, causes a crippling disabilities. Don't forget, my dear. Marked by muscular weakness, tremor, paralegia. So these are the important points. Similarly, red kidney beans, don't forget, my dear, red kidney beans and French beans containing lecithin. Lecithin is one of the phospholipids present in the nuts, and also egg yolk. What is that? Egg yolk. Okay, lecithin is also a complex lipid, compound lipid. So this is also a very, very important. Okay, my dear. So these are the important things, my dear students. Today we will finish it up to this level only, my dear students. Remaining things we will see the next classes. Okay, today what are the things already we discussed? Just we will summarize all those things. So first of all, you should note it down. What is by polyploidy, autopolyploidy, and allopolyploidy, and what are the example, critical how it becomes, that things also, and haploidy, haploid enhancement single, mutation breeding, which radiation used for the mutation breeding. Okay, what are the types they have given? Is a yellow mosaic virus is also present in the munk beans, resistant to yellow mosaic virus, powdery midway in the inducer of mutations. Mutant allergy, how many percentage of the mutant alleles are beneficial one? 0.1% only. And some examples of mutation breedings for the rice, what are they? Jagannath and Atomita. 2, wheat, Sarpati, Sonara, Pusa, Lerma. And wheat variety developed from red grind varieties such, what is that? TNIIT, Nora 64 and Lerma, Roja 64A respectively through the gamma radiation. Castor Aruna is nothing but the oil seeds. And MS Swaminathan, okay, where it located, just you note it down. This is also present in the Mumbai. MS Swaminathan lab is also located in Mumbai. And also Norman Porlag, which is a new one. Okay, lab to land. What are the properties? What are the basic principles done by these two scientists? Just about that cafeteria, concept of craft area, craft scheduling, all those things done by the MS Swaminathan, Norman Porlag. And also lab to land cultivation, that is called as hardening process, also done by the MS Swaminathan. Okay, what are the examples? What is by self-pollinated crops? What are the defects is there? What are the examples of self-pollinated crops? There are examples, rice, wheat, all those things already we discussed, particularly Vijaya and Ganga, Kalyan, Sona. These are the wheat variety. Maize variety, Vijaya and Ganga already discussed. And selection of cross-pollinated. Cross-pollinate means heterozygosity. How it will be there, the F1 generation, it will be very effective in nature. Okay. The inbreeding depression, it will not happen. All those things already we discussed. Okay. So next one is multiplication of improved seeds. What are they? National Seed Corporation is very, very important. Certified seeds also produced by the, what is that? National Seed Corporation only. Okay. And the inbreeding depressions already we discussed. What is mean? Who stated the statement of inbreeding depression? That is called a skull. SKULL. Don't forget, my dear student. This is very, very important next question. And heterosis, what is the meaning of heterosis? Already, what is the main beneficial of this one? Reverse of inbreeding depression is, what is that? Essential. Okay. It's a less harmful effect. That is it. And one more is that it is a profitable, which one, the vegetatively propagated plants such as the mango, apple, potato, sugarcane. Okay. So like that. Application of heterosis is just noted an increasing yield and nutritive value of crop plant. That is also very, very important. Increase the growth rate also. Enhance the meat production and enhance the milk yield and egg productions. And improve the performance of various animals such as the race of horses and domestic pigeons. And last one is production of new combination of colors and scents in the garden flowers. These are the important points that have been reported by the application of heterosis. Breeding of nutritional quality. The nutritional quality is also what happened. Anti-nutritional factors also present in it. Which one, some of the plants. Here, the crop produced should provide the optimum nutrition and must not contain anti-nutritional factors. Anti-nutritional factor is completely nil. Okay. What are the anti-nutritional factors? That means, about during the cross breeding, or new breeding, whatever it may be, new variety when we are discovered or introduced into the market, before that, what happened? Anti-nutritional factor completely removed. They are toxic in containing plants. As a defense against excessive grazing, examples they have given is called as So many example has given. One is called as glucosinolates, maximum present the Brassica vulneraceae. Okay, this is called as, I think you know, cabbage. Okay, cinalbumin, Brassicae, cyanogenic glycosides, alamines, neurotoxin cyanoalanines is also Lathris sativa, casseri. Casseri is not, what is a doll? Okay. Regular use of seeds, Lathris sativa, it is also one of the casseries, caused crippling disabilities marked by the muscular weakness, tremor, and paraplegia. Similarly, red kidney beans and French beans containing lecithin, these are the anti-nutritional factor present in the food stock. Okay, my dear student, that's all about today classes, my dear. We will see the remaining things on the next class. Thank you so much. Bye. Take care. Thank you, ma'am.
Thank you, sir.